Okay. So if I can encourage you all to sit down, we can begin the second session. No? That's not going to work? Can't hear me. They said they can't hear me. Okay. We'll start up. So we're about to begin the session 2, which is the session on the role of biomarkers in developing PD-1, PD-L1 combinations. And I'm David Rimm. I'm a pathologist at Yale University School of Medicine, and I'll be moderating this session. I'm going to start, unlike the other session, I'm going to start by giving a little bit of an overview of the field or where we are, which is largely not with combinations so much as with single markers, PD-1, PD-L1, and discuss the overview of immune checkpoint blockade. So these are my disclosures. So I'm going to outline the field as we are today as sort of a pretext for the rest of this session, and that is to talk about complementary and companion diagnostic tests, just what those are, because there is a big difference. And then I'll talk about the ones that are out there today in terms of categories. Immunohistochemistry, which is really the only companion diagnostic test currently, genomic testing, expression or mRNA signatures, multiplex fluorescence, and finally circulating free DNA or other circulating markers. And we'll go through each of these, and then following me, Dr. Rizvi will talk about the genomics side, or genomic testing side of things. Dr. Izar will talk about a little bit about expression mRNA signatures, amongst other things. And then Dr. Shoup will talk a little bit about multiplex fluorescence, and then we'll talk, Dr. Shulsky will talk about the challenges of all biomarker development across biomarker agnostic therapies. And then finally, we'll look forward into the future. Dr. Jobin will talk about the microbiome and cancer immunotherapy. So to begin, what is a companion versus a complementary diagnostic test? So really, a companion diagnostic test, in some ways, is where I should start. A companion diagnostic test is a test that's required for prescription of the drug. So that means that it needs to be specified in the label and theoretically is required for reimbursement for the drug as well, and actually is really precision medicine. This is the test that tells you which patients will respond and which will not. A complementary diagnostic test, which most of the tests in immune therapy have turned out to be, are not really companion diagnostic tests and arguably not really precision medicine because the FDA didn't think they were good enough to require them as a prerequisite to receiving the drug. And so complementary diagnostic tests are nice to have, companion diagnostic tests are need to have. Here's a list of all the companion diagnostic tests that are protein-based tests. It's pretty small, isn't it? There's not that many FDA-approved companion diagnostic tests as we sit here today, and this may be due to the rigor that's required for companion diagnostic testing. And as you can see, many of these are many years old. And in fact, in immune therapy, there are only 3. Now, it is hard to keep up because as recently as June, cervical cancer received the designation of a companion, not a complementary diagnostic test for the PD-L1 22C3 assay. But this is the importance of understanding the difference between the companions and complementaries. So what is these are the categories now. I'll go through each category quickly through immunohistochemistry. There are 4 immunohistochemistry tests, or 5 or 6, depending on how you count. And there are a number of comparisons that have occurred. These are 2 papers that initially describe the comparisons. And the problem was that as soon as you have a companion diagnostic test, can there be only one, or can there be multiple, or are there multiple companion diagnostic tests across a single category of therapeutics? And as a pathologist, this is a major problem for us in that clearly these 4 tests are not identical in the appearance to the pathologist, but they are all for the same category of drugs. So companion comparisons were done, and in fact, initially the blueprint on the left and the NCCN study on the right showed that there was an outlier amongst 4 of the diagnostic tests, including in the right one LDT, and then especially noted when you looked at what category you were looking at, whether you're looking at actual tumor cell expression or immune cell expression. Here you see on the lower right side the discordance of the 3 tests, even for the immune cell category. The most definitive work on this, I believe, has recently been completed, the Blueprint 2, which is a statistically powered result, which actually just came out a couple weeks ago. This was 25 pathologists from, 25 pathologists from, I think, 15 different countries, and all the pathologists were trained. It's considered a part of the qualification was to train the pathologists how to read these as opposed to the other tests, and they were trained to look at these clinical cutoffs reported in the clinical trials and to score accordingly, including both tumor cells and immune cells. But what we saw was, in fact, pathologists can agree well on tumor cells, and in fact, the tests are concordant on tumor cells, but not so much on immune cells. They were also shown to be concordant between digital slides and glass slides, and between cytology specimens and full specimens. But similar to the NCCN study, the blueprint showed the two of the assays were outlying assays, and so essentially could not be used interchangeably as companion diagnostic tests. And secondly, it showed that pathologists can't read immune cells, that even if you give them a day and a half of training, they're incapable of doing a reading of the immune cells, or at least they have concordances that are in the unacceptable range, where ICC is in the 0.1 to 0.2 range, which is unacceptable for a clinical test. I think the message here is that human beings can only do so many things, and that we have to actually look toward machines to help us to quantify things that are harder to quantify. Turns out, I think, and I won't talk about it today, that immune cells are really interesting. In fact, they're probably the cells that are driving the response to immune therapy as data is moving in that direction, including some from our own lab. But I'm going to do an overview today and not really talk about data specifically about those topics. In summary, 3 of the assays appear to be roughly equivalent for immunohistochemistry, and those are 22C3, 28A, and SP263. Cytology specimens appear to be equivalent, although not in the label. Pathologists do well reading tumor cells, and their ICC is higher when they read tumor cells than when they read immune cells, even though we're not sure that that's actually the mechanism by which the response is mediated. So that's the story on immunohistochemistry. To move to genomic testing, I think there's some very interesting new data in genomic testing. We'll hear Dr. Rizvi talk a little bit more about this. But the most recently, there's 2 ways to think about genomic testing. Genes which show inactivation of the immune system, and this reports the STK11 gene and shows that when that gene is mutated, in fact, the patients have essentially no chance of responding to immune therapy, the red line, in the survival curves. And then just the immune system and or looking at genes in a global way or a more combined way, which is tumor mutation burden. And in fact, this is the data that the best support of the tumor mutation data in the upper right-hand corner here, which showed that nivolumab in the high TMB group high TMB was predictive of response to nivolumab, and low TMB saw chemo actually benefit greater than nivolumab. However, looking forward to overall survival in that same paper in the supplemental section, we saw no overall survival difference. Perhaps most convincingly is the fact that TMB is different than PD-L1. In fact, we see almost no regression between the two. And in fact, in a series of other studies, not just the original trials, but in other work from Rizvi at Memorial Sloan Kettering and some work from Gettinger and colleagues at our institution, suggests that PD-L1 has essentially no relationship to tumor mutational burden. And in fact, the test may be additive. The best way to compare tests, however, is to look at the ROC curves, or the area under an ROC curve, and that's the AUC. And you can see that neither of these tests is very good. In fact, typically all of our companion diagnostic tests, none of them really exceed 0.7, which is just a modest companion diagnostic test. And I think it's something we should strive for, is to work toward companion diagnostic tests that have a larger area under the curve. And in fact, if you look, one of the problems with TMB is that everybody has a different definition for it. And if you thought it was hard to standardize 5 assays from immunohistochemistry, we're going to have real trouble standardizing the literally hundreds of TMB assays that exist out there. This is just comparison showing the 10% discordance between a panel, a popular TMB panel, the Foundation 1 panel, and whole exome sequencing, which already show a 10% discordance. So to summarize TMB is a biomarker for immunotherapy, and it's certainly complementary to PD-1, and it is in fact predictive of outcome. However, TMB is not standardized. This may be its biggest challenge, it may be associated with PFS and not OS, since that data has yet to mature. And then the sensitivity and specificity appears to be no better than existing tests, although the cost is probably somewhere between 5 and 10 times the amount and takes as much as 10 times as much tissue. Next thing I want to talk about briefly is mRNA signatures, and I think this is the direction that we need to be thinking about in terms of multiplexing. When you talk about immunohistochemistry, you talk about one test at a time, and what we really need to think about is how we're going to probe the immune system in a more complex way. And one of the earliest published versions of this was an 18-gene signature that was used to suggest a response to therapy, trying to look at multiple features of the immune system. And when this was done, in fact, they found that the area under the curve for this test was close to 0.75, as you can see in the upper right-hand corner here, and the combination of genes that are shown immediately to the left of it. This is now, I believe, the only published version of an mRNA signature, although I know there's a number of mRNA signatures. In fact, the Genentech mRNA signature, which is a 3-gene signature, we've heard a lot about at meetings, but I'm waiting, unless I've missed it. I've missed it. Okay, that one's also published. And actually, I think Ben Azar will talk a little bit about that in his talk. The next category I want to discuss is multiplex fluorescence, and this is a category of interest where instead of looking at multiple RNAs or multiple DNAs, we look at multiple proteins. And this is a category of interest to my own lab where we look at, we try to do high-plex analysis and then distill that down to perhaps a few genes that can be seen in multiplex fluorescence. And this is a way to, we can not only assess the tumors, but assess the microenvironment at the same time. And this is some data, some early data from Tumay et al., that in 2014 showed the difference between that patients that responded had high CD8, and Roy has talked about this, and I'm sure you're all aware of it, that it's the microenvironment where the T cells are that may need to be probed to find the most information. In fact, this is some data from my own lab, the only data I'll show from our lab, where we looked at melanoma quantitatively with multiple quantitative CD markers and other markers, and show that in fact, CD8 is highly predictive of response, the hazard ratio of 0.29. And in fact, this is the only data I'll show on combination therapy, where we looked at, in this melanoma cohort, got either Ipi-Nivo or Pembro or Nivo alone. And you can see that the area under the curves here with quantitative fluorescence is approaching 0.8. In fact, on the lower right-hand corner, the predictor for dual area under the curve for dual Ipi and Nivo was 0.83, although all these have AUCs in north of 0.75, which I think is hopeful predictive value, and I look forward to other data from other people doing quantitative fluorescence to try to look at the microenvironment and multiplex the microenvironment to try to get more information here. This is another example of multiplexing protein group that was looking at PD-1 and PD-L1 at the same time, and when they co-localized, or HLA, DR, and IDO, when they co-localized, was shown to have predictive value. And this fluorescence co-localization study that's done is coming out in clinical cancer research, was presented at AACR, but is about to come out in clinical cancer research. And the last category I want to just briefly touch on is circulating free DNA or other circulating markers, because wouldn't it be great if we didn't have to do a biopsy and we could predict response or progression on the basis of circulating markers. And this is probably still in its earlier stages. Perhaps the best data for this is the preliminary data from the BEFIRST trial that Vamsi Valchetti presented at ASCO just recently, and you can see the patients with high TMB. This is not TMB of the tumors, this is TMB that was found in the circulation, is showing, these are the dark lines over on the right side here, showing that the subgroup with the best response has the dark blue lines below the curve. Notably, there's a lot of patients that have TMB that's less than 16, less than their cutoff, also showing response. So again, this is still early data, and we look forward to ROC curves to compare these tests to other tests. But I think certainly worth keeping your eye on. And then finally, maybe we don't even need to look at the free DNA itself. We can just look at actual other variables in the blood, like neutrophils and lymphocytes. And this is the Lippi score, the NLR score, where they looked at the neutrophil lymphocyte ratio and found that this is in fact predictive combined with as a combination score. So this is just sort of an introduction that I wanted to do to the area, a very brief introduction to where we are. And then we'll now have each of the speakers talk about those areas that I outlined, but then discuss overall this whole area of biomarkers and some of the key issues related to the biomarkers during this session. So, Nair?